So, welcome to our first podcast from Scientarius Institute. Today, we want to talk about a very old picture, a very old paper from about extremophiles. But first, we want to introduce ourselves. So, I welcome Nick. Hello, Nick.
Hello, I am Nick Nielsen. And?
Yeah, and I'm Christoph Lahtz. Yes, hello. So, yeah.
The paper we're going to talk about today is some comments on the evolution of extremophiles by R.D. Macelroy.
Yes, and from when is that paper? I think that was before 1970, right?
1974. It's a 1974 paper.
Oh, from not. Oh, 1974. Yes, yes. Okay.
And it includes 4 references, with the papers in 1970, 70, 73, 71, and 73. So only 4 references. Very short paper, 5 paragraphs, a page and a quarter. And this is the first paper to introduce the concept of extremophiles.
Yes, what is noted here, the Archaea bacteria were not discovered until 1977. So that is 3 years before that. So people really didn't know about this totally other branch of living organisms, which are, most of them are very
extremophilic.
Yeah, okay. I guess we get better over time, but okay. So let's dive right in. It's not very long.
So he starts with mentioning thermophiles, psychrophiles, which are cold tolerant, and halophiles, which are salt tolerant, and he notes immediately that these organisms are known, and there's many papers around them. And actually, I found a paper from 48 or 49 that shows the term mesophile was already being used in the late 40s. But then he says that proposing the name extremophile as a descriptive for organisms to populate environments hostile to mesophiles, or organisms which grow only in intermediate environments. So there's a recognition that there are organisms, mostly very simple organisms living in extreme conditions, but he's the first one to put them all together under one heading. And he notes that there isn't any known common mechanism of origin, but he also says that we may discover related mechanism may eventually be demonstrated to account for the diverse tolerances found to exist in nature. At least that was the hope when he wrote the paper in 74.
Mm-hmm. Yeah. Yeah, the whole comment is about that, to see if there's a common ancestor of extremophiles, or if there is an adaptation, right? And it is not clear, or it was at least in that, if I understand that right, in that paper, it was not clear at that point if extremophiles were the first organisms and then developed to normal mesophiles, or if that was first mesophiles and then or extremophiles. I think I understood that in that way. Is that correct?
He actually, in this short paper, he actually offers both possibilities. So in the second paragraph, he suggests that extremophiles evolved from mesophiles. So he says, quote, they evolved gradually from ancestors which grew only in intermediate environments. And then in the fourth paragraph, he suggests the alternative, that mesophiles evolved from extremophiles. So there's 2 different possible evolutionary routes here, and it's not known as he's proposing this category. But we still don't know. That's one of the unknown questions of the origin of life, and it's not necessarily one or the other, because there might have been a class of extremophiles that evolved first and issued in, evolved into mesophiles, but there might also have been at the same time something like mesophiles that eventually evolved into extremophiles, because life on Earth is long enough that you could actually have an oscillation between extremophiles and mesophiles. And also, depending on how you define extremophiles, the earliest life is going to be extremophilic simply in virtue of the fact that it's not going to be like life today, because there was no oxygen, there was no photosynthesis, and all the things that we think of as defining mesophilic life today, the conditions didn't even exist.
Yeah. I mean, I don't know when they discovered the black smokers and the life down there, but that was way after this. So they didn't know about these extreme environments without light, where actually it's the whole ecosystem, right? So
yeah, there's whole ecosystems have since been discovered since this paper came out that weren't even guessed at. What do they call the kind of is it thermosynthesis that is, or chemosynthesis is the term that's used for that black smoker life.
Oh yeah, I think chemoautotroph, right? I think that is what they use there, the bacteria. Yeah. So
he also notes here in the last paragraph, fifth paragraph,
oh.
while no unique unity has been discerned yet among extremophiles, and the assumption of a common mechanism is doubtful, it can be proposed that the weak intramolecular forces upon which biological systems depend may be the key to the understanding of the functional and structural stability of macromolecules in extremophiles. So he's proposing, just speculatively, what may be a common molecular origin of extremophiles. And I don't know if any of that research has been followed up in the sense the paper was published, or if it's even in any sense a research program to look for what he calls functional and structural stability of macromolecules of extremophiles.
Yeah. Well, yeah, I mean, it depends what kind of extremophile it is. For example, thermophiles, right? So there was the whole cell membrane is differently structured, of course, because it needs to be more stable and more liquid and higher temperature. That means the melting point is higher. When it goes back to normal temperature, it's more stiff, so it cannot exist there. So that's why I was actually a little bit confused by the comment he makes here in this long paragraph when he says here, thermophilic bacteria enzymes have been found to be, with few exceptions, functional at moderate as well at high temperatures. So thermophilic enzymes work in high temperatures and low temperatures. So I was not sure that works, because that is true. I was a little bit confused because
isn't that what we now would call thermotolerant?
Could be, yeah.
Now we
Maybe.
make a number of distinctions between, thermophile and thermotolerant, or halophile and halotolerant, being, the difference between having to have those extreme conditions merely to live versus being able to survive them, but not necessarily thrive under those extreme conditions.
Yeah, you might be right when he writes here thermophilic bacterial enzymes. Yes,
with few exceptions. Thermophilic bacterial enzymes have been found to be, with few exceptions, functional at moderate as well as high temperature. They might not simply have been as much knowledge of thermophiles at the time. There certainly wasn't as much knowledge of thermophiles at the time. though it might be interested to, statistically lay out what percentage of thermophiles or perhaps even hyperthermophiles also are thermotolerant and can survive at the less than optimal temperatures for them.
Yeah.
As you say, they have particular adaptations that make it possible for them to live at high temperatures, but it doesn't work so well at lower temperatures. And at some point, it's going to stop working, and they're going to die, just like if we're going to stop working and die if we get in too high a temperature.
Yeah. So our proteins denature or lose contact with the amino acids, basically lose the shape of the protein, because the energy what they get introduced because of the heat kind of denaturate, and then it's going to change. You see that when you have egg white, that's basically a lot of protein, and then you put that in hot water, and then it becomes totally denatured that it comes from. But extremophile enzymes and proteins, they are much more stable, so they can tolerate a lot more energy intake before they fall apart. So, but that makes them so stable that when they get colder, they are basically frozen. For them, it's like a freezing point. So, and then they cannot act anymore. So, yeah.
Do they go in
simple terms, yeah.
Do they go into a kind of hibernation, or do they die from that?
No, that's just they just cannot do their function anymore. It's almost like they are frozen. It's not really hibernation. I don't know. There are probably some extremophiles that can go in hibernation like that. I mean, when you look at the Yellowstone lakes and stuff, they are hot, and that is true. But there are probably areas where you can take a little scoop of sand and you can find extremophiles in there. So when you put them up in high temperatures, so yeah,
then they'll start growing at high temperatures.
Yeah, they can only grow and multiply and have a metabolism in high temperatures. Yeah, there are probably some that can go in a hibernation, like in a spore or something like that, but they cannot function. Yeah, in high temperatures. Yeah, you're right. Thermophilic, that would explain that. Instead of, the whole terminology is not laid down in this paper, so that makes this paper kind of the first try to explain all these things, which is very interesting.
And as we've discussed on several occasions, and not recorded, but there isn't yet, even decades later, any systematic taxonomy of extremophiles. There's a lot of attempts, but in discussing extremophiles, for example, the Extremophile Handbook lists 14 different categories, you could call them, of extremophiles. But since that's been published, there's already been several other suggested kinds of extremophiles. There are extremophiles that grow in virtually non-nutritive environments of low or absent water, or low of absent air pressure, et cetera, and you can just keep piling them up. But we don't have any way to really organize our knowledge of extremophiles. So when Macelroy says that he's basically introducing an artificial idea to include these things, but he's hopeful that there might be a common mechanism to explain them, but we haven't come up with a common mechanism, and we need to think more clearly about why we're calling things extremophiles and why we divide them into the categories we do. So here in this first paper, we've got the recognition of three different kinds: thermophiles, psychrophiles, and halophiles. And like I said, the Extremophile Handbook recognizes 14. There's more ideas out there for extremophiles, and probably more ideas will be forthcoming.
Yeah. An interesting side note, I want to say, I didn't send you the paper, and we probably will discuss that on one later video, but there is a paper about that the early Earth was probably purple instead of green, because halophiles were all over the place, and they have rhodopsin, I think, and that is purple. So they did photosynthesis. So there was a certain halophile. So that was really interesting to see that the early Earth was probably purple and not green before photosynthesis with chlorophyll was developed.
So this was an earlier form of photosynthesis, distinct from chlorophyllc photosynthesis. Is that what you're saying?
Yes, yes. They use a different photon catching mechanism. So yeah, so the rhodopsin, I think that was in the paper, but we will discuss it on a further note, and on a later point. Yeah.
I did hear something about purple Earth, but I haven't read the paper yet, but I did hear something about that. I think it's Robert Hazen in his book, one of his books. He's written many books. He divides the ages of Earth into various colors. Blue Earth was when there was it was all covered with water, and there were no geomorphological features that could interrupt the global ocean. But then you get, I think, red Earth from the banded iron formations, and all these colors come and go over geological periods of time. So another interesting feature of this Macelroy paper is the earliest life they mention here is 3.35 billion years before present, and one of the papers cited in this in the four references here is the Chemistry and Morphology of Precambrian Organisms by Brooks, Muir, and Shaw, in which they talk about a volcanic deposition, more specifically, relatively unmetamorphized sedimentary rocks of the Ondrevakt group. So from what is now Swaziland, or what was Swaziland. I recently noticed that country was renamed. So this is what was known in 1974 of the earliest clearly identified early life fossils on Earth, fossils of microorganisms. But since this paper has come out, we have older examples, 3.5 billion years before present, in an Australian chert, and there's even been some more speculative work on some crystals that have been found in Quebec that may go as early as, just after the formation of the Earth. So when you have these evidence from different periods of the Earth's geological history, you might form, a particular view of the origins of life, and then as that gets pushed back, you might have to revise your views about, whether it all started with extremophiles or mesophiles or something that can't be defined as either. As I said earlier, by definition, the earliest life on Earth would not have evolved under mesophilic conditions because those conditions didn't exist yet.
Yeah, that's why, but that is speculative, right? That the first life was very likely extremophile. So, and it will come back, in a few billion years or so, or maybe because of climate change a little bit earlier. So the extreme conditions will come back, but yeah.
I've actually been working on this quite a bit lately because the whole planet is going to undergo directional selection as it gets hotter. So as the sun begins to expand, even before it enters a fully fledged red giant stage, the sun is slowly growing and getting hotter. And so over the next 2 to 3 billion years, the Earth is, while it's still habitable, is going to get hotter all over continuously. There still could be interruptions. There still could be additional ice ages if the sun goes into a solar minimum of extreme proportions. But overall, over the geological period of time, we're going to have directional selections toward thermophily. And so it's going to be, a very interesting contrast to the earliest history of Earth, where you, like you said, you may have had extremophiles in the past, and we get extremophiles in the future, but in the future, they will definitely be organisms evolved from a much more complex biosphere than existed in the earliest stage of Earth life. And things evolved from mesophiles today, contemporary mesophiles under directional selection for thermophily in 1.5 to 2 billion years from now could become something very different.
Yeah, maybe even multicellular life or extremophile life. Yeah, like the tardigrades, for example. Maybe that is a good. But I don't know how. And I think they are
the charismatic megafauna of extremophiles.
Yeah. Yeah, they are cute. What really is interesting, what I read, that thermophiles actually live also in your hot water tank. So, and I never tried that out, but that is worth an experiment to do that if there are really extremophiles in there, doing boil that with some sugars, and then let's see sequence that or so. So that's very interesting.
But you would have to keep it hot, right? If you wanted to study them. If you extracted
water,
yeah.
You have to.
If you extracted water from your hot water heater, you'd have to keep it hot as long as you wanted to study those.
Yeah, you have to keep it hot. That's very difficult to grow. There are also some nothing to do with the paper, but there are some hints that there are bacteria from so deep from the out of the Earth's mantle. They divide so slow that you cannot grow them in the lab, and you cannot even identify them as life, because they just don't grow. You have one cell maybe somewhere, and because they don't divide that fast, because there's not much nutrition. So you cannot culture everything. So I mean,
is it a case of extreme pressure or lack of nutrition, or both?
Good question.
Once again,
could be that the pressure is not there, and then they basically die, because they would basically pop, right? Because they have a high internal pressure to counterpart the outside pressure. So I don't know. So that could be one case.
You'd have to have a very specialized laboratory apparatus to keep the pressure and keep the heat on continuously, and then find a way to grow them. This is one term I just ran across in the past year or two, is the scale problem. And I've been seeing, as I think about it, I'm seeing this in more and more places, that certain things can't be done at a different scale than they exist in nature. So for example, gravitational confinement fusion. You have to get fusion from gravitational confinement, you have to have something as big as a star to produce enough heat and pressure in the center that it creates nucleosynthesis and fusion, and then radiates energy outward. We can do fusion by inertial means or by magnetic confinement. We cannot in a laboratory, unless you have a laboratory bigger than a star that encloses the whole thing, you can't have gravitational confinement. So it's a scale problem. What you're saying there is there's a scaling problem both with these organisms that are deep in the Earth's crust. There's a scaling problem with being able to do that in the laboratory, because we probably can't keep the heat and the pressure on for a long enough period of time to grow them in a lab, because they divide so slowly. So it's a scaling problem. Yeah. Partly a scaling problem in time, and partly a scaling problem in terms of the apparatus that would be necessary in order to grow those in a laboratory. So that defines a kind of limit on the research you can do.
Yeah, what you could do is you could put the lab down under, basically, right? So you could have a very simple module, I would say. So what has a sample of the environment, and then grows there, and then you can lift it up, or sequencing right there. But yeah, again, under pressure, things work very, very differently. Yeah, so we talked about to have basically in a zoo black smokers or so. That will also probably not really work. We would have simulate that with something else. But yeah,
but
it would be nice to see.
Interestingly, I've run into at least a couple papers where there have been limited simulations of something like black smokers in laboratories. Obviously, they don't work at the same pressures as under the sea, but there have been limited attempts to do this in a laboratory. So it might be worth looking at.
Oh,
so,
oh yeah.
So some people working in origins of life have been doing this.
Yeah, cool.
But
yeah.
Once again, it's partially a scaling problem. You could do something like a black smoker in the laboratory, but you couldn't do something exactly like you have in nature. So it's a question of, well, the other term that's used in science, usually used in physics, is symmetry. So it's symmetry of scale. Is there a symmetry of scale? Symmetry of scale usually breaks. If you do an experiment of one size and then you increase it by an order of magnitude, it usually doesn't work, or decrease it by an order of magnitude. But translation in time or translation in space, that is to say, to do something at a different time or do something in a different place, often that does work in physics theory. So these are translational symmetries, you could say. And the question is whether if you had symmetry in scale, then you could do something on a small scale and it would apply to the larger scale of the actual world, the small scale of the laboratory, that is.
Yeah. Yeah, it's a super different, super interesting topic. That's why we follow that with extremophiles, because you can use extremophiles or the enzymes for so many different things, and you can look out for how would life be on other planets. So one simple very important enzyme, what we all use now because of the COVID pandemic, is a PCR, right? So the poly chain reaction, what we have, and this enzyme, what is able to the polymerase, what is heat stable, comes from an extremophile. I think even Yellowstone Park. So and this little enzyme was responsible for so much of scientific progress in the molecular biology area. So that is really it would very important. So, and there are probably so much more enzymes we could use for science and scientific experiments. So it's a kind of a gold mine, these enzymes, and also to look up to how is it on Mars, on Venus, on other celestial bodies. Can there be something? What is, or is there a condition which is somewhere on Earth is the same, and we know there is life? So then we could say, okay, the condition is the same because there could be life. If life developed there, that's a whole different story. But we could say that life what we have here right now could survive in that condition over there. So yeah, so we could think more about, that's a little bit more science fiction, but about terraforming, what extremophiles we would need, or what organisms we would need to make the,
by matching up extremophilic niches on Earth with similar analogous extremophilic niches on other worlds, then we could start creating a program for ecological succession on a bacteriological scale, microbiological scale, you could say, for other worlds. So you start with the extremophile that could, in theory, live in this environment and introduce it. and then from there, that's your seed of life, as it were. From there, you grow toward more mesophilic conditions, you could say, until more and more terrestrial type organisms could be added to that mix and go beyond merely the initial extremophile toward a more complex ecology.
Yeah. A very interesting organism or effect here in this way is, I talked about that earlier in some talks, that these are, and I think you told me about that, these are thermogenic plants who produce heat.
Yeah, we did discuss that at one time.
Yeah.
And that's something Dyson talked about in his later years, of that the way to go in terms of making habitable environments off the Earth was to send these thermogenic organisms to asteroids or other astronomical bodies, let them take root there and grow, and then manufacture their own environment that would, and then guide it toward something being friendly to human beings. Then you'd have another place human beings could live in theory.
In theory, yeah.
But since we're talking,
you would need to.
When you're talking about terraforming, you're talking about something on geological scale. So once again, that's a scaling problem. So if just like I was saying either earlier in this conversation or before we started recording, that you could have one extremophile starting on in the Western Australian chert, and another extremophile starting in the Anvarok sediment in Swaziland. And even though they're separated by 700 million years of time, they still could have existed as separate biological communities in the early Earth until the Earth became a honest to God biosphere, in which you get the they're biologically integrated in one ecology. And so say the scenario we were just talking about, where you have extremophilic niches on another planet, then you would say you seeded those, but there are multiple ones, say multiple extremophilic niches on Mars or Venus or something like that, and you seeded them over geological time, then they could grow in the same way. Before, it would take a long time to cultivate a biochemically integrated biosphere on a world that's so far from habitability as these. So you could get very different forms of life evolving from these places that you've seeded with extremophilic organisms.
Yeah, they could build different domains, like the shadow domains, what is kind of an idea here on Earth. So a little bit for explanation, there are shadow domain is a domain of life what we don't recognize as life because it's so different, and we don't find it because we don't look for it. So I think one example was I think NASA was that, right? They found an organism which could interchange phosphate for arsenic in the DNA, right? But I think that was not always that could interchange these two molecules if the conditions were in the right, in the right order. Then it could happen. So that normally it did phosphate, but it could switch it out. That something like that. We wouldn't look for that, or some life what is a little bit different than, or maybe different base pairs, which is already, that would be crazy too if you would find that. But
or maybe you know it, but you don't know it's a shadow biosphere, because that would be a way of thinking about, before the Woese 3-domain model of life on Earth, people knew there were archaea, but they didn't call them archaea, and they didn't think of them as a fundamentally separate domain of life from bacteria and eukarya. So you could think of archaea as a shadow biosphere before it was recognized as part of the terrestrial biosphere. That's not the way it's usually talked about, but that would be one way to think about a shadow biosphere. But we will
Yes,
in
that's true.
future,
Yeah.
we will in future discuss some papers on shadow biosphere and go more into that because it's an intrinsically interesting biological idea.
Yeah, that is true. Yeah. So I think just a comment for this paper. It has no experiments. Just it's a comment on the evolution of extremophiles. Very short and very different than paper from today. Normally you have an abstract in these papers today and then a conclusion. And this paper really doesn't have that. It's just
It's more
talking
like
about this.
It's more like what we would call a survey paper today. So instead of presenting original research, it's a survey of the domain and offering a new concept to use in further research. That concept being the concept of extremophiles. So that's why we're picking this paper to talk about first, is because it introduces the idea of extremophiles. So, people knew about thermophiles, people knew about halophiles, etc., but nobody had, before Macelroy had, put the concept, this umbrella going over these other concepts to join them together.
Yes, that is true. And where did you get this paper? Is it accessible so we can put a link in the description or something?
I don't remember. I'll have to look up. I'll have to look up where I got it and see if it's easily available or obscure, and then we can at least put a link to the journal from it where it's from. Well, it was I think it says here on the corner.
Yeah, you will.
Yeah, I don't know. It doesn't.
We will put a link, or maybe even we can upload that on the website, something like that, if that is possible. Yeah. Okay, I think we discussed everything. Or that we can discuss more, but that would be different topics.
We will be picking up classic papers in extremophiles, astrobiology, origins of life, and bioastronautics, which are the four research foci of Scientarius Institute. Our tagline being life in space and extreme environments. That's what we're interested in. That's what we want to talk about. So yeah, thanks for listening to our first, perhaps a little awkward talk, but hopefully we'll get better at it.
We get better. Okay. Yes. Okay. Thank you for watching.
Thank you very much.
Bye-bye.